Hello and welcome back. We're going to be discussing two main topics today, protein synthesis and central dogma. Central dogma is kind of the overall idea that we're going through, but the ultimate purpose of this is to produce proteins. Learning outcomes for the day. We have three main ideas, and yes, this will be kind of a long video, so just hang tight. We'll be here for a while. The main learning outcomes for the day. Let's define what a gene is and what genetic code is and explain the functions of genes. We'll also be naming two phases of central dogma and describing the roles of the various pieces, including DNA, messenger RNA, transfer RNA, and ribosomal RNA in these processes. And then I want you to be able to compare and contrast three main players. Those would be triplets, codons, and anticodons. What exactly are these? And really the key is to know which these belong to, because they belong to the players in the second learning outcome. Triplets are actually the pieces of DNA. Codons are the sections of messenger RNA, and anticodons are actually pieces of transfer RNAs. Now, this next idea goes back to the time when Francis Crick and James Watson first discovered the structure of DNA, and we, at that time, began to understand how DNA does what it does. DNA, or your genes, are effectively a recipe for a protein. So, we need to do a two-step process to get proteins from the original instructions genes, and that process begins with copying down the original instruction in a process known as transcription. That gives us a temporary copy of the gene in a piece of molecule known as messenger RNA, or little mRNA. Once we have that copy of the instructions, we can then get the final protein, and that final protein is made at the ribosome in a process known as translation. For me, this whole central dogma two-step idea is very reminiscent of something near and dear to my heart, and that would be something that a lot of you may have access to at home, some of your old family recipes. My grandmother used to make cookies for me when I was growing up, and that, for me, was kind of a nice fundamental, founding, basic idea of childhood happiness. So what I have left of that at my age is my grandmother's old cookbook, which I unfortunately don't have. It's at my mother's. She won't let me have it. She says I can have it when she dies, but you know, I'm not in that big a hurry to have her die, so it's fine that it's with her. But I do still like, when I go to visit my mother, to open this old book and copy out favorite recipes. I can't take it away with me, but I can certainly break out a card and copy down the recipes that are favorites. And one particular favorite of mine is my grandmother's cookie recipe for snickerdoodles. A lot of you can probably appreciate that. So what I would do, if I wanted to make a batch of those cookies and I didn't have the recipe handy, I would go to my mom's, I would open the book, and I would copy down that set of instructions. Then I would put the book back on Mom's bookshelf. Now I've got the card. Can I tear up this 3x5 note card into, say 2 or 3 dozen pieces and eat the little pieces of paper and enjoy my grandmother's cookies? No. Of course, I've got to follow these instructions and actually make those cookies. And at that point, after having followed the instructions properly, and I assure you I can make these cookies great, then I have the end product. So just like the original recipe to the copy of a recipe to the cookies, DNA to RNA to protein is very much the same thing. The original archive of instructions, DNA, stays in the nucleus and is not allowed to come out. Messenger RNA is a copy of that recipe, and the end product would be the protein. So keep in mind, this is a two-step process from DNA to messenger RNA where you copy down the instructions. That's called transcription. And by the way, the word transcription means to write it down. Translation is where we take those instructions and then do something to interpret them into something useful, and translation in this case gives us proteins. So that would be central dogma, the idea from Watson and Crick that DNA is instructions to make proteins, and we have to go through the intermediary of messenger RNA to get that done. DNA is a master blueprint for making proteins. A gene is a section of DNA that has the instructions to make one polypeptide. Effectively, a gene is a recipe for a protein. And triplets are how we read DNA. Triplet, a 3-base section of DNA, is effectively a word, a piece of information in the making of a protein. Keep in mind that DNA is made of bases that are A's, G's, T's, and C's, and each group of 3 describes a single amino acid in a growing protein. So a triplet is 3 neighboring DNA bases. Might be GGG, might be TAG, might be CGA, could be any combination of those 4 base types. I do need you to take a little time when we're done here and go to the study area of Mastering A&P and watch the animation on DNA and RNA, and that's going to be something that looks vaguely like this. When you get to the study area, go to chapter 3, scroll down a little bit because this picture was taken a little farther down the screen, and then you're looking for the animation DNA and RNA structure. Take a little time, watch that. You don't have to do it right now, but before you stop studying for this, please take time to do that. Moving on, protein synthesis. Genes are interesting things. They're not good information front to back. They contain useful information and extra information. And those would be exons and introns. Exons are the actual pieces of the gene that are the instructions for making a protein. Introns are non-coding segments. Now, this used to be called junk DNA. It's not really junk DNA. It's just simply not involved in describing how to make the protein. Imagine looking at a magazine and you're flipping over to, I don't know, page 24 to an article you want to read, and you read the entire page 24, including looking at a couple of pictures on there, and you get to the bottom of the page, and it actually says continued on page 79. If you turn from 24 to 25, your story doesn't continue, does it? You've got to skip the intervening pages until you get to, what was it, 79? So those pages between 25 and 79 in that magazine would be like introns in the gene. Now, we'll talk in a few minutes about how that bit gets edited out of the messenger RNA, because indeed it does. RNA has several roles in the whole process. It's a DNA decoding mechanism, and it's a messenger, and it's also a transporter. There are 3 types of RNA that are all found in the nucleus, and they're all involved in making proteins. And those 3 types of RNA would be messenger RNA, ribosomal RNA, and transfer RNA. And we'll look over the next few minutes at the role of each of these in making proteins, because they all have their own role to play. RNA is different than DNA, keep in mind, because it's both single-stranded and it does not have any thymines. Instead of thymine, it's got uracil. So just keep in mind a few of the differences between DNA and RNA, because we're going to talk about those a lot over the next few chapters. Let's talk briefly about the 3 different kinds of RNA and what generally they do. The first is messenger RNA. It's abbreviated little m, capital R, capital N, capital A. So messenger RNA is effectively a copy of our original gene. It's got all the instructions we need to make a protein, and it is sometimes described as a complementary copy of the gene. In other words, back in the DNA, if there was an A, this is going to have a U. If there was a G, this is going to have a C. If there was a T, this is going to have an A. And I'm not going to keep repeating those because you can kind of get the point. So messenger RNA is a copy of the instructions. It is the recipe that will be used by the ribosome. By the way, we'll get there in a minute, to make to make protein. The next type is called ribosomal RNA, and ribosomal RNA literally, actually, physically is a part of the ribosome, which is this factory that makes proteins in the cell. It's a structural piece of a ribosome, and the picture that just appeared on the screen is a ribosome. By the way, this ribosome in this picture is reading some messenger RNA right now. The messenger RNA in this picture is purple, and the new protein that's being made is that string of blue beads. So ribosomal RNA is a functional, structural part of the ribosome. Let's take that ribosome over to the side a little bit. Keep it in mind because it needs to do something for us in a few moments. And let's talk about the third type of RNA that's involved in making proteins, and that would be transfer RNA. It's abbreviated little tRNA. These bind to amino acids. They pair with bases on the codons of messenger RNA at the ribosome and kind of help with the process of protein synthesis. They, in fact, serve as delivery trucks to deliver amino acids to the growing protein. Headed over to the ribosome, it's bringing in the amino acids we need to make the new protein. So in kind of a graphic summary, here's the deal. Here's where all this stuff happens. We have a cell on screen, and inside the nucleus is where transcription takes place. We have our original DNA that is copied out to messenger RNA, and that needs to be edited, removing the introns, and then finally the exons leave the nucleus. And by the way, that's how I remember this. The exons, the good bits that actually have all the instructions, are the part that actually exit the nucleus, and they leave out through the nuclear pores. Where they're headed is to the ribosomes, which exist out in the cytoplasm, and the ribosome out in the cytoplasm will then read that messenger RNA, and from that will make a protein. We'll look a little closer at the process, but this is a good, simple graphic summary of the entire process of central dogma, which includes the 2 steps of transcription and translation. Protein synthesis actually happens in 2 steps. We've talked about this whole summary already. This is just kind of putting in text on the page what you and I have said over about the last 7 or 8 minutes. Protein synthesis happens in 2 steps, transcription and translation. Transcription, just as a reminder, is DNA being encoded into RNA. Messenger RNA is effectively a temporary copy of the gene, which then gets read by a ribosome and made into a protein. By the way, polypeptides, fancy way to say protein. All right, 1 step at a time. Let's take apart the 2 main pieces of central dogma. Transcription. This transfers the information that we have in the gene into a temporary complementary copy of messenger RNA. Complementary, again, by that I mean that where we had a C in the DNA, we have a G in the RNA. Where we had a T in the DNA, we have an A in the RNA, and so on. There are several factors that help this happen. They're known as transcription factors or gene activators, and these are chemical molecules that actually help get this process started. They loosen the histone or packing proteins from the DNA, getting the area that needs to be copied opened up and ready. A promoter area is where we're going to bind these. And they'll also mediate the binding of the enzyme that actually does the copying. The enzyme that reads DNA and makes RNA is called RNA polymerase. And I'll show this to you graphically in just a minute. Transcription happens in 3 phases, and they're kind of the obvious things that you would expect. It starts, it goes on, and then it stops. The starting phase is called initiation, and during initiation, RNA polymerase, the enzyme that's going to do the copying of the gene, binds and then actually separates the DNA strand in an unzipping move. Then we have elongation. During elongation, the RNA polymerase is actually running down the length of the gene and making a copy of it. Once the RNA polymerase gets to the end of the gene, there actually is a little stop area, kind of like a period at the end of a sentence. Then we have termination, and we stop at this point. And it stops when the RNA polymerase gets to a particular spot on the gene that indicates a stop should happen. Human messenger RNA actually needs to be edited, and it needs to remove the introns, because remember, only the exons tell how to make the actual protein. The introns are the extra stuff. Again, used to be called junk DNA, not a good term anymore. And those introns are removed by sections those introns are removed by enzymes known as spliceosomes. Now, if you're going to be taking microbiology or you have taken it in the past, you need to have a special little reminder here. Bacteria have no introns, so they skip the spliceosome step. There are some proteins that are associated with messenger RNA to help guide it out of the nucleus through the nuclear pore, and these also help to guarantee its accuracy for translation. Here on this page, we're going to, kind of a piece at a time, look at the process of transcription graphically. Now keep in mind this is all happening on the DNA, which means it's happening inside the nucleus. We start out with the molecule RNA polymerase finding, recognizing, and binding to the promoter region. Think of this as sort of the front porch area just in front of the gene. The gene also has a termination signal at the far end, and that's going to be the place where the transcription stops. Once the RNA polymerase reaches there, it will stop copying DNA. As we start here with initiation, we have some transcription factors that help the RNA polymerase bind. The RNA polymerase is unzipping the DNA at this point, and it's beginning to make our copy of messenger RNA. As we come a little farther down and look at elongation, the RNA polymerase is moving off to the right in this picture, and the copy of the RNA, known as an RNA transcript, is being made, and it is effectively just a copy of the DNA. As we reach the terminator, or the termination signal, RNA polymerase will be released and the messenger RNA is completed. At this point, we are done and we have a copy of the gene. Now focus in on the details of what's going on. Here in this zoomed in picture at the copy site, we have the RNA polymerase that is both unzipping and reading the template strand of DNA and then making a copy of the RNA. There on that DNA, every place where we have a C on the DNA, we'd put in a G on the RNA. Every place we have an A on the DNA, we put in a U on the RNA, and so on. So this little zoomed in area shows you kind of nicely exactly what's happening as that RNA polymerase runs down the length of the gene, making our messenger RNA copy. Translation effectively converts the base sequence of the RNA into a protein sequence. Now this will happen on the ribosome, and it involves three kinds of RNAs. It involves both messenger RNAs, transfer RNAs, and ribosomal RNA. The genetic code is the three base sequence of DNA represented by codons on the messenger RNA. A codon is a complementary three base sequence of RNA, and that sequence is actually representing individual amino acids. That sounded kind of complex, so let me show you how to read the genetic code. Generally, the genetic code is represented in chart form, and as we see here, it's a big chart that has codons plus amino acids on it. As you look down the areas that are lighting up now, these would be the codons. They are the complementary matches of the triplets that we saw in the original gene. So these match up, A for G, G for C, and so on, what we saw in the original DNA. These are codons, and they are sections of our messenger RNA. So if we took a long strand of messenger RNA and sliced it into bits and pieces, these are the letters we would find: A, U, U, A, C, U, C, A, A, and so on. So what I would like you to do is to understand how to read this genetic code chart. What we have is a whole list of codons which then tell us which amino acid to put into our growing protein. For instance, if we look at CUU and then follow the bracket off to the side, we know that the amino acid we need to put in the growing protein is leucine, or abbreviated LEU. Look to the right just a little bit, find CCC, that's our codon. Which amino acid goes in place if CCC is our codon? That would be PRO, or proline. I'm going to stop highlighting, and I want you to find ACU. Now, I'll leave it unhighlighted for a minute. You find ACU, and once you spot it, notice which amino acid would be next. Yep, you found it. ACU is right here, and the amino acid that would call for would be threonine, or THR for its abbreviation. So that's how you read the genetic chart. Let's try one more before I go on and talk about the next bit here. Find CCA. CCA, you got it? It's right here, and the amino acid that it would call for would be proline. This is how you read codons and find the appropriate amino acid. Now, I want to take just a few moments and point out a few particular codons because they have some special properties that we need to be aware of. The first one is highlighted in green here, and it's AUG. AUG is often called the start codon because that's what the ribosome recognizes and binds to as it begins to make a protein. If we approach the analogy of a piece of messenger RNA from a gene being like a sentence written out in English, then the way you would typically start a usual sentence is you capitalize the first letter. So you can sort of think of AUG as that capital first letter in a sentence. It's the way you recognize the beginning. with that analogy in mind now, how do you recognize the ending of a sentence? There's a punctuation mark, something like a period or a question mark or a exclamation mark that tells you you've reached the end of a sentence. The end of a piece of messenger RNA ends in one of three possible stop codons, either UAA, UAG, or UGA. So those four you probably actually want to memorize because they actually have some major significance. So that's how you read the genetic code, and you may well see a question on an exam asking you to read a sequence of messenger RNA and produce an appropriate protein from that sequence. Now, transfer RNA, there are actually 45 different transfer RNAs, and they bind to particular amino acids. These, again, as a reminder, are like the delivery trucks we spoke of earlier. They're delivering particular amino acids to the ribosome to be built. They bind each specific amino acid at one end of the transfer RNA. The opposite end, apart from where the amino acid is carried, is called an anticodon, and it actually has matching bases that match up to the appropriate section on the messenger RNA. And the typical binding pattern that we see whenever we look at nucleic acids, C's to G's and so on. So if the codon of the messenger RNA reads AUA, the anticodon, which is a part of the transfer RNA, would read UAU. This assures that they match up appropriately at the proper spot. Ribosomes coordinate the coupling between messenger RNA and transfer RNA, and the ribosome actually has three docking sites on it. The first one is called the A site, second one the P site, and the third one the E site. The A site is named for the aminoacyl peptide, P site for peptidyl site, and the E is the exit site. There are three phases of the translation event that require ATP, protein factors, and enzymes. There's initiation, which of course is where we begin producing a protein, then elongation, where the protein gets longer, and finally termination, when the protein is finished and the ribosome releases the finished protein. We're going to take a look at each of these in turn, beginning with initiation. The way initiation begins is the small subunit of the ribosome, it's actually a two-part structure, finds the AUG, the start codon, on the messenger RNA and grabs onto the messenger RNA at that point. The large subunit will join after the small subunit has found the messenger RNA, and that creates our functional, ready-to-work ribosome. At the end of initiation, what we have set up is the transfer RNA is in the P site, the A site is vacant. Elongation has three separate steps. There's codon recognition, during codon recognition, the transfer RNA is going to bind the appropriate complementary codon in the A site. A peptide bond will then form. The amino acid of the transfer RNA in the P site is bonded to the amino acid of the transfer RNA in the A site. Graphics will help with this, so if it sounds a little confusing, just wait till we get pictures up and it'll help. And then we finally have translocation. The transfer RNA will shift over one spot from the A site to the P site and the P site to the E site, and then the one that is in the E site exits. That's why it's called the E site. Elongation continues as new amino acids are added at each step, and the initial portion of the messenger RNA is read. And effectively what we form here as time goes by is a structure known as a polyribosome, which has multiple ribosomes reading a messenger RNA at the same time. This actually can produce multiple copies of a protein simultaneously. A polyribosome looks like the image that you see here on the screen. On the left in the cartoon image, we can see that multiple ribosomes can read down the length of a messenger RNA at the same time, and as each of them in turn reaches the end and finds the terminator site, it breaks free, releases its protein, and goes on about its business. And then each of these in turn, as they're moving off to the right here, will reach the terminator and release. The picture on the right shows a polyribosome. And again, a polyribosome is a single long strand of messenger RNA in all of the multiple ribosomes that are reading it at any given time. Termination happens whenever a stop codon gets reached by a ribosome. And again, those stop codons, just to remind you, were either UGA, UAA, or UAG. Again, kind of like with a sentence ending in either a period, a question mark, or an exclamation mark. When the ribosome comes to a stop codon at the A site, that signals the end of translation. The protein is released from the ribosome, and water gets added to the chain. The ribosome comes apart, and ultimately, if we're done with this messenger RNA, it's broken down. The protein gets processed into a nice, fully functional, ready-to-work protein, and that's often finished in the Golgi apparatus. Now, graphically, all of what we've said over the last few minutes looks like this. I'm going to step through this one piece at a time, so we'll put this summary together. Don't worry about all the details right now. Let's graphically take a peek. What we see here is in the upper left-hand corner, our DNA has been copied, and the copy is messenger RNA, which comes out the nuclear pore. It's found by a ribosome, and again, the ribosome starts grabbing onto messenger RNA at the start codon, the AUG, and notice this first transfer RNA has bound to the start codon, and it matches appropriately. U matches A matches U, and C matches G. That first transfer RNA is carrying the amino acid methionine, and the large subunit assembles onto the small one at that point. Look in the A site. What's bound to the A site? Nothing yet, but the next codon is lined up. It's AUC. And the appropriate transfer RNA a little bit later on will join us right there. This is the step we would call initiation. We have just gotten ready to start making a new protein. The small subunit has bound to AUG on the messenger RNA, and the large subunit is attached. Let's go on to the next phase. Here at the next phase, we have everything lined up, and the next codon, CCG, has been lined up in the P site. The empty A site has a CUA, and it's waiting for an appropriately matching transfer RNA to land, so that would be the one that's coming in here, GAU. When that binds, the next amino acid in our growing protein would be leucine. You can see it being carried in right there on the transfer RNA. So what we have going on there is codon recognition in our elongation. The codon, in this case that we're matching up, is CUA. The anticodon GAU on the transfer RNA will match with that perfectly and bring in leucine to be the next amino acid in our growing chain. As that happens, we create a peptide bond between the new amino acid that was just brought in and the protein that we've already begun growing. So right there in red, you can see the brand new peptide bond that got formed. What's going on here is, in effect, our ribosome is acting as an enzyme and lining up the pieces to produce a new bond, and that's what you see happening right here. As this progresses, the entire ribosome shifts. In this particular case, it'll be shifting off to the right, and that will free up the transfer RNA that was previously in the P site, moving it to the E site. It's empty, so it actually gets ejected. I kind of like to use those E's there, don't I? The empty one moves to the E site and is then ejected. That leaves the A site open with the next codon we need to match. Our codon here is CUC. Can you predict what the next anticodon on the incoming transfer RNA might be? CUC would have to match it, so it's going to be GAG. We don't see it here, but that's what will be coming in. When we get to a stop codon, there are 3 of them, and it looks like we've just brought UGA into the A site. That's going to carry with it a release factor, and at that point what's going to happen is the entire protein is released, and again we get the entire summary here. We completely release that protein, the ribosome falls off, and we've completed making our protein. That's the process. Over the next few pages, we'll take a look at the role of rough endoplasmic reticulum in protein synthesis. Remember, what makes rough endoplasmic reticulum rough is the fact that it is covered in ribosomes, the very little guys we've just been taking a look at. So let's talk about how the ribosomes and rough ER work together. First of all, messenger RNA ribosome complex is actually directed over to the rough ER by a particle known as the signal recognition particle, or SRP. This is going to dock the ribosome onto the endoplasmic reticulum, and then the growing protein will actually enter the endoplasmic reticulum as it's formed. When it enters the endoplasmic reticulum, a protein may be modified in various ways, including having sugar groups added to it. Its shape can be altered, and once it's finished, the protein will then be packaged into a vesicle, a membrane bubble, for transport over to Golgi apparatus. So let's look at that graphically and kind of walk our way through that. Okay, we got the good summary image here. We're going to take this apart and do this one piece at a time. First of all, the signal recognition particle, the little gray guy here, helps to dock the ribosome onto the rough ER. When it's docked, the protein begins growing into the rough endoplasmic reticulum. The protein then finishes. Along the way, we may have sugars added to it. We can see that happening right here. And when the protein is finished, it's released into the space, the cistern of the endoplasmic reticulum, and it's then packaged into a transport vesicle and pinched free from the endoplasmic reticulum, and then floats away. This, by the way, is a coated transport vesicle, and that's going to be then stored in the Golgi apparatus. So we have a summary from DNA all the way to proteins, and this is again central dogma. We have complementary base pairing, which directs the genetic information flow from DNA triplets to messenger RNA codons. Once that's done, the messenger RNA codons will match up with transfer RNA anticodons, guaranteeing us that the right amino acid sequence gets put into place. And the anticodon sequence is identical to the DNA sequence, except for the fact that we've got uracil substituted for thymine. Graphically, all of that looks like this. We start with our original gene, and that gene is made up of triplets. So groups of 3 bases in the DNA we would call triplets. Those will be complementarily matching to the messenger RNA codons. The messenger RNA codons themselves are complementary matching to the anticodons on the transfer RNAs, which carry the appropriate amino acids to make our new protein. That was a nice long run through a pretty complex topic. Thank you for your time on this, and I will see you in our next video, which will be considerably shorter. See, I almost forgot to check and see if you got this stuff. Before we leave, concept checks. Did you get these points? Triplets, codons, and anticodons are all 3 base sequences. How are they different from each other? How do the A, the P, and the E site of the ribosome differ functionally during protein synthesis? And what is the role of DNA during transcription? Now, really, this time. See you later.